Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARK Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARK. All statements made regarding companies or securities are strictly beliefs and points of view held by ARK or podcast guests and are not endorsements or recommendations by ARK to buy, sell, or hold any security. Clients of ARK Investment Management may maintain positions in the securities discussed in this podcast.
Hello, welcome to FYI, the For Your Innovation podcast from ARK Invest. I'm Brett Winton. With us today, we have Nemo, who covers AI and drug discovery for us in the multiomics space. Hi, Nemo. How are you?
Great.
Yeah, we have a great show today. Nemo and I talked to Alex Z, the founder and CEO of Insilico Medicine, which is a AI drug development and discovery platform. Nemo, lay out the AI drug discovery landscape for us. Kind of what is the promise here? How are these companies approaching it? What are we seeing in the marketplace? And then where does Insilico fit into kind of your understanding of the space?
Yeah, so the big picture kind of promise here with the AI drug discovery is that, AI, it's pretty good in seeing certain patterns in the data or generalize, if there is enough data, and like that kind of gives us the result. And so, when we think about generally in the kind of whole space of drug discovery, where we could see the most kind of interesting kind of application is in this kind of hit to lead and the lead optimization kind of part of the drug discovery. So this is the part where you're actually trying to design the drug and kind of optimize the drug. So for example, if you take antibodies as the example here, so you need to kind of find the antibody that is binding to your target, but that antibody, in addition to binding to your target, also needs to kind of have certain other qualities to be actually a really good drug. So AI can actually really help you do that kind of multi-parametric optimization and kind of optimize the different parameters at once, versus you in a traditional kind of lab, in traditional drug discovery, doing each step once at a time. The second part here is that, for the antibody, for the binding, if you run a big enough experiment, your model can learn from that experiment, basically the bigger space, and then for the next kind of campaign, you can be actually faster in developing that, finding that antibody that's binding your target of interest. So that's the kind of the most kind of common area where it's kind of used. They use it, or we can also use it in the target the discovery and validation. In preclinical stuff, you'll see now what happens with organoids, but this is the most common area.
Yeah. So basically, there's this whole front end of the drug development process. It could, in traditional space, it might take you 5.5 years to do it. And to get a drug or enough drugs through that whole process into clinical trial with enough candidates that are going to fail out into clinical trial that to actually get a drug to market, it might cost you $280 million traditionally to just get there. And the promise of AI drug discovery is, hey, all of this stuff that we're doing before we go into human, we can use, rich data sources, potentially automated kind of experimentation driven by your AI models to develop, better binding, more potentially novel, like going after unique biology. But even if you understand the biology and everybody's going after it, you can develop something that gloms onto it better, and you can get to market, faster, cheaper within a drug candidate that, though this is yet to be validated all the way through by anyone really, but should have a higher odds of actually kind of delivering the successful outcome that you want, because it binds better, even though you spent less kind of like time and resources getting to market. That's the promise.
Yes, and I would say that, as I said, this kind of the preclinical phase kind of has the three kind of target, designing the drug, and the preclinical stuff. Designing the drug is the go-to area. The target ID, I think there are people doing that, but And yes, I think there is some like low-hanging fruit that you can just mine the like literature that exists. I think the right way there is really, we don't understand enough of the biology. So it's really kind of multi-omic orthogonal kind of data sets where you're kind of combining, perturbation, single cell, and then combining that with the patient data to actually find the new targets. I want to go to a preclinical, side, preclinical, I mean, the issue, when we are trying to kind of make the IND studies, there kind of, again, the whole issue that there is a lot of failure is because, we will do these first two phases, for example, in the 2D cell culture. Then this preclinical stuff is in vivo and we do it in mice. I mean, obviously, and then you're training your mind to another system, and then from there you're going to humans. And so the preclinical phase kind of, that kind of part of the discovery has a huge drop, but I think it's more because the models are bad versus, it's something else.
By the models being bad, you mean not the AI models, but actually the mouse is a bad proxy for a human.
Yes,
it's you can solve cancer in a mouse, and that might not solve cancer in a human. And in fact, you might fail to solve the mouse cancer, even though you would have solved the human cancer if you were able to skip the mouse step. And so that's another area where we need to improve things.
There's a joke. So one of my PhD advisors, he's a very famous mouse cancer biologist. I think he made the first mouse to study cancer. And so there was like a running joke in the lab, like more than 100 times we can cure the cancer in mice, but it still didn't do it in humans. So there's a huge difference, right? Yeah.
So, and then at an aggregate level, at least the way we think it's going to evolve is that to get a drug to market traditionally costs you, including all of the things that fail and go wrong along the way, around $2.4 billion and takes you 13 and a half years. We think, and there's reason to believe this is actually a conservative estimate now, given how the FDA is enabling, faster steps along the way, that with AI, you can get a drug to market for $600 million. So call it 25% the cost in 8 years instead of 13 and a half. And perhaps even less. So that's the promise of the industry, is that you spend less money and take less time to bring, something that's going to help people to market. And so therefore your ROI just is much higher. It's, closer to kind of a technology company ROI, whereas right now industry-wide return on R&D is pitiful. It's around 4%. So in example companies going after this are Recursion and AbSci in the public markets. There's companies that are still private, including Insilico, going after this. How does Insilico's approach, as we'll learn about, differ from some of the other companies that we've seen and looked at in the space?
Basically what they have done, they kind of try to put different models from start to finish. And so for this kind of first piece of the kind of target ID, they kind of aggregate a lot of different publicly available data sets and also kind of research papers, publications available, and then basically try to kind of stitch the different models together to kind of mine that area and find some of the potential targets that they can go after. And then after that, they have the kind of chemistry, they call it, I think, chemistry 42 or something like that, which is their kind of generative kind of platform. And so which is kind of multi-agent kind of autoencoders, basically kind of trying to, find the kind of molecule, which is the small molecule for that particular kind of target. And then kind of also when they're doing that, kind of trying to optimize the, as I said, few other things which are important for the tox studies. So that's kind of their primary approach. So it's really about much more being in silico, how they say it, and then they do the kind of validations later.
Yeah, in contrast to, say, Recursion, which seems to focus, give me the contrast.
Yeah. So and obviously I'm a fan of Recursion, so just, disclaimer. But so Recursion basically kind of took, makes the kind of biology and chemistry maps. So whole idea is that there's biology is very complex, and for us to understand it, we're gonna, use the kind of try to map it in 50 different cell lines. So kind of to have kind of different kind of biological kind of contexts, and then do basically different perturbations with either with CRISPR to remove the genes, or we do it the small molecule. And then we're going to do readouts, both kind of phenotypic. So we, look at the kind of imaging of the cells, but also get the molecular one. And like that, we are actually, trying to really understand biology better and then actually come up with the new targets.
Yeah. And so the way I think about it is, you could imagine, is it better to train a model on all of the academic literature that's out there? Or is it better to train a model on the underlying biology. And Recursion's approach seems to lend itself more to, hey, we're going to enable automatic experimentation on the underlying biology and have the model get an understanding of the biology by studying that data directly, as opposed to we're going to take a vast body of academic literature, some of which is valid, some of which is flawed, and try to annotate it to like just identify the good papers and then train it on those papers. And of course, both companies are using some combination of approaches, but it's where kind of capital and allocation and resources are really going, right?
Yeah. And just to be also, Insilico, I mean, they also take publicly available data sets, right? But like the problem is that all of that is so small. And so again, like each, it's Recursion also, like it's a cell culture, right? Which has issues on its own, right? So, right, like always there is some issue, but to me, the Recursion approach gives you more kind of, if you pair it with real world data, which they are doing with Tempus and the other ones, then you're actually I'm getting here very deep biology signatures, which I can go to patients and look for them there. And then therefore I can, maybe validate the target. Versus kind of, and there could be also when you do the kind of literature search and all of that, there could be some low-hanging fruit, right? So, I mean, the Future House just came out with, right, the molecule. It is like more, but that's more like, the repurposing, I would say, but.
I mean, I think the way to think about it is the industry standard right now is so pitiful that there's a lot of like, basically doing an intelligent, aggressive approach of devoting AI and ML techniques to both existing data and to help you generate novel data should broadly be a way to be able to, develop drugs more efficiently. And there's reason to believe what's provocative to me about this space is there's the potential that, one or two players kind of gets a nose ahead, which then gives them this flywheel of data generation and kind of validation that then allows their underlying model to grow very large. And so then the consolidation in the space moves towards kind of whatever this innovation engine it becomes, as opposed to right now, the very big players in the space are basically have massive distribution. They're basically sales companies that take drugs and, and they have a franchise in oncology or they have a franchise in cystic fibrosis. And that's the mechanism by which the industry is consolidated. There's the potential that like actually the underlying discovery platform driven by AI becomes the consolidation mechanism of the industry. And if you look at the big companies in the industry, these are worth hundreds of billions of dollars easily for big companies. And so you could end up with an AI drug discovery franchise that is worth hundreds of billions of dollars. That's could occur. I think that's an easy possibility to foresee. Not to say that one or the other is necessarily going to do it, but that's the promise here is you have a better return on R&D company that then uses that advantage to generate better data for itself, which then improves its return on R&D. And so it kind of stays ahead of everybody. And we'll see.
Yeah. And it could be also the kind of, a lot of the small molecule stuff, is actually not that great as drugs. Where when you think about biologics, is that the kind of cell gene therapies, which are kind of more the cures or like antibodies, they're much more precise, and specific than the small molecules. And then, especially if you think about the, cell and gene therapy space, right? You're basically, your drug there, like there is you have to deliver it and then it has to kind of act on. So you're like basically decoupling kind of specificity, where the drug needs to act and what it needs to do. With a small molecule, you have that issue all the time, right? So that's where I feel that kind of more on the biologics kind of side, which kind of takes all of this, antibody cell gene therapy, that's the way to definitely to go.
Well, in some ways it's required for truly personalized medicine to do it biologically, right? A small molecule is something that like it plugs in all over your body in all kinds of different ways, right? As opposed to like, I have this particular key that I'm designing for this particular lock that I know I specifically want to turn.
Exactly. I mean, and that's like why antibodies, and also I think the small molecules are actually not that amenable for the AI, generative AI. Why antibodies are so amenable? Because antibodies as proteins, right? The solution space that AI is learning on is already kind of constricted, and there's like a sequence, the DNA sequence versus the kind of final antibody. There is a kind of relation between that, and so like that, you can your machine learning AI can actually learn and kind of develop new ones. With small molecules, you don't have something like that, because we can make anything. So that's why I'm very skeptical about that side. And then obviously the gene editing, right? I mean, or the mRNA as well. Like it's sequencing is binding to a sequence. It's a very specific, the gene editing as well. So it's just much more precise in the specific, I would say.
Well, so enjoy the conversation with Alex. We get into the end. We get into some really meaty discussion about longevity. So make sure you stick around for that, particularly if you want to live forever, which I'm not sure everybody does, but I think a lot of people do. So enjoy the conversation, and thanks for joining FYI, the For Your Innovation podcast. Hello, welcome to FYI, the For Your Innovation podcast. Welcome, Alex Z. I really hesitate to even try to pronounce your last name, but it's, you can read it on the screen, and I have done it successfully once. Alex is the founder and CEO of Insilico Medicine, a talented scientist. Alex, lay out kind of who you are, how you are, where you are today, and where Insilico is, please.
Sure. So today I'm serendipitously in Newport Beach at a conference where I'm presenting, but also it has multiple partners here. So we do get to speak shop. I am a scientist by training, computer scientist. I did my first 2 degrees in computer science at Queen's University in Canada, worked in semiconductors, made enough money to pursue my lifelong dream of kind of chasing longer lifespans. So everything I do is focused on aging research. I don't care about anything else. Did my grad work at Johns Hopkins, worked for a number of biotechs. 2014, at the cusp of the deep learning revolution, started Insilico to apply the power of deep learning to understanding life itself and going after aging. So we actually were born out of the NVIDIA GTC conference, which is kind of a nice touch. Since then, we've nominated, so we operated as an purely algorithm-focused company till 2019, and then released our software to the market, so people use our AI. I also use the same software to discover drugs, and then sell those drugs to the pharmaceutical companies, so licensing them out. So that's kind of our main business model. And since 2020, we've nominated 22 developmental candidates. So that's kind of one notch before human clinical trials. 10 reached clinical stage, and 1 has completed phase 2A efficacy study in humans in idiopathic pulmonary fibrosis. So I'm very deeply proud of that. We try to publish as much work as we can. So we nowadays publish at the rate of about 1 paper every 6 days. In good specialized journals. And we try to open source some of our software to make the community try our tools and get community level validation. Very focused on aging, and I recommend all of you who is listening to start thinking about life with that angle. So because whatever you do in life, whatever money you have, whatever you have achieved, is going to gradually disappear and possibly completely disappear. So why don't we unite and fight this great enemy together?
So Insilico starts in 2014, focusing on using AI in drug discovery, or at first it was just like AI biology and understanding longevity, and then you moved into drug discovery. Is that the transition that occurred?
So originally we started as a purely algorithmic company. So we were applying deep learning specifically to many different tasks, many different problems in the pharmaceutical industry. We started as a target discovery company, so trying to understand which protein drives the disease and also is implicated in aging. And usually that's just not one protein, it's a variety. So we were trying to understand the intimate interplay of those proteins in time, in aging and disease at the same time. Turned out, at that time, the pharmaceutical companies, and even today, they are not exactly interested in target discovery. So target sciences is a very important field, but most of that is performed in academia. And academics do not have huge budgets to partner with AI companies. So we had to pivot and develop chemistry expertise. So we started working with pharmaceutical companies to work on their own targets, on their own proteins, to develop specific chemical matter that could make a good drug. And that's where we started utilizing in 2015, 2016, generative adversarial networks. So that's what we were famous for. And then transitioned to generative reinforcement learning to design molecules with the desired properties. So basically, instead of looking for a needle in a haystack, we started generating perfect needles and then selecting out of those needles, the perfect one. And then pharmaceutical companies challenged us to a very large spectrum, very broad spectrum of problems. Some of those problems were clinical, some of those problems were biomarker discovery. So we were pretty much like a sous chef for the chefs in deep learning. And then we realized, so then since we have this entire Lego blocks of algorithms that could be fit together in order to cover the entire drug discovery and development pipeline, we actually started thinking about pulling the trigger on our own drugs. And that's so until 2019, we did not touch our own drugs.
Got it. But it seems like from inception, 2014 to now, gosh, has the AI landscape shifted dramatically. I mean, attention is all you need. The introduction of the transformer architecture happened, I guess 3 years after the start of the company, and that's unlocked so much. Generative adversarial networks, I remember that was the way to do image generation. Now that's all done on diffusion. How has Insilico reacted to kind of like adapting in these new tools? And then where do you sit today relative to, it's like, oh, you had a tool that was working. It seems like in a lot of cases, it's like the new tool is probably more potentially performant. Like, how does that happen?
Absolutely. So nowadays, my greatest challenge is balancing the advances on the AI side with the kind of lengthy validation cycles on the drug discovery side, right? So it takes you sometimes a few weeks to develop a super intelligent model, but then it's going to take you several years to see if it can produce molecules where the generation conditions are confirmed experimentally. So you need to actually wait all the way to humans. So whatever you see right now in clinical trials that we have, and some of those molecules are performing extremely well, is the result of what we had in 2020, 2021, 2022. Now, of course, we are accelerating the validation cycles. But in terms of the technology evolution, what I figured is that in our industry, in AI-powered drug discovery, not always you need to be at the absolute cutting edge with the most frontier model. What is important and ultimately important is experimentally validated models. So if the model can perform a certain task extremely well, so basically you tell it, okay, predict toxicity in this cell line and in this organism. If that model can do it really, really well, looking at the molecular properties that you are giving it, or the biological hypothesis, or both, it's good enough, right, for this specific task. If you can show that is done it over, let's say, 50 times, it's like having a very good validated expert in your team. Doesn't matter whether the expert is old or young or whatever, right? So as long as they can do the job, I really like, Deng Xiaoping's quote, it doesn't matter whether the cat is black or white, as long as it can catch mice. So if your model can really do the job, you will employ it at some point in time, because I can tell you that currently,
you might have a black and white cat and then you're overrun by rats and it can only catch mice, right? And so, but I think is the particularly like, is it useful to have a very predictably performant model for internal purposes, or is that particularly useful in kind of like partnering? Because then you can go to somebody external and say, look, I have all of this evidence that the thing works. And so then maybe it fits into the kind of like business logic of kind of like licensing drugs and in a better way than saying, I have the most insane Ferrari of a model here, but you just have to trust me, it's going to work.
Yeah, so Brett, you actually originally touched on the most kind of sensitive and the most important topic in AI-powered drug discovery, right? So how do you keep up, right? And I think that we're still on this subject. How do you keep up with the most capable models that are coming up, right? And you can be outcompeted by rats, right? So I just wanted to make this kind of punchline. So it's extremely important to have the most frontier model that we can have constantly in development. But you need to have a long tail of models that you trust, that you can fall back onto, that are validated. So you have this entire zoo of models that work, that are validated, that you trust, that your medicinal chemistry team trusts, that your biology clinical team trusts, and also your partners trust, right? They know it works. So they can always fall back to something that is validated on previous generation, but you are always working on the frontier one. I always learn from SpaceX. So the company that I deeply admire, and of course, I have Elon's picture everywhere. Whenever I think that I am, working too hard, I'm looking at this picture and I realize that I'm slacking. So look at how they are building rockets. So you have the Falcon series that works extremely well. So you get the payload to orbit and beyond, and it works. But at the same time, you're working on Starship, right? And you're working on next generation Starship already. It might not be as validated, right? You wouldn't put passengers on it, but you will be working on Starship, and you are using the learnings from Falcon in order to get a better Starship. So that's exactly what we are doing in drug discovery, because at the end of the day, you need to develop ultra high quality drugs. This is where you cannot fail. It's kind of like space with passengers, but there are more passengers. So you cannot harm them, and you need to get the payload to orbit. So in order to do that, you need to have very validated model architecture with many models. You need to be able to run simulations using those models. You need to be able to ensure that the entire market trusts these models. That's why you also publish. But you are always developing the next Starship. And once the Starship works, it becomes part of the Falcon series that's validated. You're working on the next Starship. So what we are doing at Insilico now, I like to use the term pharmaceutical superintelligence. And by that, I mean prompt to drug. So if I can explain what I want, the system would make it for me, and then I will start my validation cycle. Currently, it takes me around, and please don't kind of judge me on that or quote me on that. That's a ballpark estimate. Whatever I say now, don't buy, sell, or take any drugs based on what I say, please. So usually it takes, the computational part takes us about 3 weeks, right, for any launch. And to get to phase 2A complete, just to get like basic signal of efficacy in humans without like big claims, it takes you about 5 years. So out of this 5 years, if you can get 3 weeks if you compress it to a week or a day, which we also will do, the rest of the validation cycle, the experimental work that you will need to do, is vastly longer and vastly more expensive than the computational part. So you better have a validated system. It doesn't matter if it's 3 weeks or 4 weeks or 5 weeks, right? As long as it works, because the stakes are very high.
Yeah, I had a follow-up question. So you were saying, about the models. It's about kind of trusting the model, and so kind of model showing you that in this cell line or in this model you can, there is no toxicity, or what is the toxicity. But like, how do you deal, we know that cell lines most of animal models, they're not good. And so how do you, like, if your model is predicting in those instances, how do you ensure, like, for the kind of clinical trials and the human side?
Yeah, so the only way to prove to yourself and to the others that your model works, and usually it's not just one model, it's an ensemble of different models, it's a pipeline. Of course, we're turning it into now multimodal, multi-species, multi-omics, super capable models, transformer diffusion, many other architecture types. At the end of the day, you need to perform the experiments. And your first date with a molecule starts with binding, right? So you synthesize it, you test it for binding affinity, and that first date will cost you about $2,500 to $5,000, right? And about like 2 to 3 weeks. Then you, once you establish that the molecule is a good binder, and you test many other properties, so you see if it binds to your molecule, protein target and nothing else, then you put it into cells, right? And see if it performs a certain function and results in a certain a response in cells, right? And that's your second date with a molecule. And then you have many dates before you commit to a committed relationship. So then you take it to a prom, right? And you put it into mice and see whether the mice metabolize this molecule the way you predicted it to get metabolized. You see if it has any secondary metabolites, if it degrades. You see if it hits something that you don't expect in an animal. And that's your kind of prom, right? To get there, usually it takes you about a couple months on average, and it will cost you a few $100,000, right? Because before that, you will have to take a few other molecules to dates, right? So usually we synthesize about 60 to 200 molecules per program. So you need to basically test a few. Usually it's kind of like, imagine if you want to get married, but all of your potential candidates are like Miss Universe contestants. So how do you know, right? So you need to do a little bit longer validation. I'm sorry for using this analogy. I hope nobody is offended. It's the first time I use it publicly. You basically need to take those kind of models to proms. And I don't date. It's just, sorry, it's just an analogy. Then you basically need to see if it works in a disease. So this is the most important challenge. So basically figuring out which animal model or human-like model, organoid, or maybe some ex vivo explant, right? So you take primary tissue from the real patient and test something there, or collaborate with an academic that has a really good model. So which model is most representative of the real disease, right? So if we had that for like most cancers, we would already solve those cancers. But currently, none of those models are perfect. So you usually do 2 or 3 or 4, and then you once you demonstrate that your molecule worked in several of those models, you commit to a marriage, so to speak, right? And that marriage is called developmental candidate. So after that, you just have the formal testing. So then you move with the speed of traffic with what regulators want you to do. So you do the formal toxicity testing. It's called IND enabling studies. Usually that is 28-day GLP, so good laboratory practices, toxicity studies in 2 species. That usually puts you down about $1.5 to $3 million, right? And takes 9 to 12 months. I wish I could skip this step. So far, we have not failed in those studies, but you have to do that. And then you apply for IND to start formal clinical trials. And by the way, that is your first year of marriage, right? You still don't have kids. And by the way, I don't have a family, so I don't have kids yet. I don't know how it feels. So please don't be offended, making an analogy out of my mind. And that process from like 0 to developmental candidate, when you kind of get married, usually takes us on like typical is around 13 months now. And shortest was 9 months, longest was 18 months. And to answer your question, I know I took a long route. Like, how do you know that the models worked? So you basically build up the validation databases, right? So the more launches you have to developmental candidate, the more evidence you generate that your models work. So far we developed 22. It's basically like, again, we learned from SpaceX. SpaceX is the best example for drug discovery. So you basically say, okay, well, I did 22 orbital launches and developed payload, right? So now I know that this model is validated. And some of those models you can update, you can include the new ones. But if the AI drug discovery company, if they claim to do AI drug discovery, does not show you, let's say, 5 or 10 developmental candidates out of the model, I would not trust that model. And I would not trust that AI drug discovery company.
You can look at Insilico's pipeline and say, well, you have 2 assets at phase 2 or 1 asset at phase 2 and a couple at phase 1.
So we have 1 program that is in 2 phase 2s, yeah. And 1 completed phase 2A.
And so kind of like traditional space, it might cost you, adjusted for failures, $400 million to get into phase 2. I think Insilico,
you know your drug discovery.
Yeah, Insilico has collectively raised around 400 million. I don't know exactly how much you've cumulative raised, but that's
just more than $500 million. Yeah.
Okay. So 500 million, but there's at least evidence that you've gotten something to phase 2 with, I'm sure you have some cash in the bank. So with around, $400 million plus everything else that's in the pipeline, which is all of the IND enabling. So there is in some ways, Nemo and I are always talking about and kind of approaching skeptically. It's like, because when AI is moving so quickly and it's so capital attractive right now that every company in the world is being urged to and is incentivized to say, yes, we're using AI. We're using AI to manufacture iced tea. We're using AI to, whatever. Any business needs to have like an AI spin in order to raise capital and attract attention. And, often that's like a layer of AI frosting on top of an existing business. So within at least there's some evidence that Insilico is bringing drugs to market more efficiently. How do you think about kind of the idea of, a frontier monolithic model versus kind of call it bits and piece models that you're piecing together? And then in what direction is your platform moving to, using another Elon company analogy, their autonomous, their robotaxi software was a whole series of some ML, some traditional coding, some more software 2.0 or AI driven components that over time they've consolidated into. It's still not a single model, but it's closer than it was.
It's getting there, yeah.
Yeah. Where are you? I assume that's the direction that the industry is going to go. So one, is that true? And then where are you on that journey? And how do you even track how to allocate resources towards accelerating that or just kind of pacing along at that advance?
Yeah, very good question. So, and really good analogy with robotaxi. So because it's very, very similar idea and also similar to SpaceX, right? Again, that's why we all love Elon and learn from him. So I've invented a really cool concept that is new, and that's mine. So I call it the pan-flute reinforcement learning. So you basically have many specialized smaller models. Now we have around 700 of them. I know that each one of them in certain settings works extremely well. So it's basically like a small expert in one small domain. And very often we just stack them together to achieve a certain goal, right? So basically you create a Lego building out of those small Lego blocks. But you can also take those small models and rank them by their computational cost, right? You say, okay, well, this one, small one, it basically just looks at the molecule and says, is drug-like. It looks like a drug, it looks like it's okay, right? So it's not trash. So basically hot dog, not hot dog. Remember the Silicon Valley series? But it costs you like peanuts, nothing when it comes to computation. Then the next step would be, oh, does this molecule have any liabilities from the toxicity standpoint here and here and here? What's the probability it's that is going to degrade, right? What's the metabolic stability prediction? And many. So basically create this pan flute, but every time you're including a new model, you're putting it to the right in terms of computational complexity, right? So at the end, you basically have very complex, expensive simulations. And let me just finish. So now imagine that you have a super intelligent generative model that is trained on all kinds of data: proteomics, transcriptomics, omics, chemistry. So I love this. So in 2022, DeepMind published a really powerful concept called Gato. A lot of people ignored it, but it's a generalist agent, one model that can perform many tasks. It was largely ignored because within each task, it could not perform extremely well, right? So you would still rely on specialist models to perform those tasks. But it had mutual information. I said, guys, let's drop everything. We're going to use this pan flute approach to train very capable multimodal models with our own small kind of zoo of validated models. So you train your generative model on all data you've got. Sometimes maybe it's not super high quality data, but then you massage it, you teach it how to generate ultra high quality molecules, right? So basically it plays with the smaller expert to create drug-like molecules. Then it goes to a more computationally expensive expert and learns how to understand toxicity. And then it basically distills the model that understands folding, docking, affinity prediction, generation with the desired properties. And then you go to 3D, and then you go to clinical. After this process is done, your multimodal model becomes super capable and basically distills the expertise from all your pipeline. And that is something that we are most focused on right now. So you're basically transitioning from basically Falcon to Starship. And that mega model is the Starship. It's one model that can do it all, very versatile. And usually now we do it in biology and chemistry separate. So we have biological models that do this, right? So highly capable, that understand life in multiple species. And we have chemistry models that understand chemistry. Now we're playing with the concept of uniting them, even though most of the time you don't need to, into one model that can do super intelligence without the need to rely on smaller models. But in a drug discovery cycle, usually you're medicinal chemists who are very used to, let's say, some of your 3D models. They would always go back and validate computationally some of their hypotheses using those smaller models, right? Because it doesn't cost them as much as the real experiment, and they get significant kind of peace of mind that the model is going to work. So you are working on something that is super frontier, next generation, but at the same time you have the zoo of models that can serve as employees, right? So they can perform a certain task, or they can serve as teachers for larger models. And you know that they are validated. They went through multiple experimental cycles, so you can trust them, and you can trust them to train larger models. So that is the trick.
Is the right way to think about it, though? It's almost like, I like the pan flute analogy. Like, you have like a bunch of just different tubes and you're training some kind of player of those tubes. But some of those tubes probably aren't I mean, there are probably more traditional machine learning type models that are underneath, right? Like it's almost like tool use where you have an orchestration agent that's you are reinforcement learning about using the right tools to produce kind of like the outcome that you need. How do you think about, and what is the like data mix that's going into that training? How do we think about kind of the richness of data that you need, kind of the potential orthogonal data sources that you need to tap, and kind of like the data off internal experimentation versus data collected from, I don't know, patient samples or anything else? Like, how do we think about that?
So the vast amount of data that is used for training nowadays and also for testing is publicly available data from experiments that have already been published. And I kind of started cutting my teeth in AI-powered drug discovery by, believe it or not, automatically analyzing government grants in the US, right? And then around the world. So we track funding globally. And we track about $2.5 trillion worth of funding. And then you kind of clean up this data, right? So you maybe bring it down to like $1 trillion or less. So you cut maybe like more than 50%. And then you put it into buckets. So biology, chemistry, text, clinical data, further refine it, apply human experts. So in our early days, so 2014 to 2019, we hired many like PhD students, master's students that cleaned up the data, annotated the data using human intelligence, sometimes kind of scored these similar papers. So there could be two different papers published on the same protein telling different stories, right? Because experimental results were different. Which one do you trust? So first of all, you need to kind of highlight those conflicts. And sometimes, again, the protein moves in a completely different direction in two different experiments, right? In similar experimental conditions. So you need experts to validate this experimental data. Sometimes they don't do a good job, but they highlight the problems. And then you link the publicly available text data to publicly available omics repositories, chemistry repositories, and train on that, clean it up. And that is the main. So I actually think that if you cannot train on public data and demonstrate that you can deliver a PCC from that data, you should not be an AI. You are just a traditional biotech, right? So if you need some additional data, usually it's a good excuse. Oh, what am I lacking? Oh, I'm lacking an expensive machine that I will show to my investors. That's how I'm different. No, like, look, ChatGPT, Grok, like, what kind of unique data do they have, right? I mean, it's Twitter, X, and many other sources. It's publicly available. So if you wanted to, you could actually collect that as well and clean it up and create a super intelligent model that understands the universe, right, to some extent. And if you think about yourself even, right, so you didn't read all the books in the world, right? You just need to read the good books and understand the first principles of certain processes. So that's what we are doing. We're using publicly available data, but later you need to have some golden data sets that you trust, that you generated yourself, in order to validate whether this publicly available data is good or not, right? And whether your model works. And that's where we have a massive automated laboratory, 6 interconnected, fully automated rooms working around the clock with minimal human intervention that are generating validation data, right? But again, I just want to emphasize this is a bonus, right? So that lab can help you identify new targets, do indication expansion, do toxicity analysis, generate really good quality data to see if your model worked, and faster. But compared to the publicly available data, it's like, right? So you need to do the groundwork, you need to grind the data, And if you didn't grind the data available to you publicly, you should not exist as an AI drug discovery company.
I think the argument would be that like imagine that there's a river that you need to cross. Being the first person to build the bridge across the river is extraordinarily valuable and actually dissuades the second person from crossing the river if you've crossed soon enough, right? And so and so even knowing how to bridge build, like you still need to build the bridge. And kind of you still need to adapt to the terrain of the river bottom to successfully build the bridge. And the final bit, the reinforcement learning that you're doing to your engine that allows it to get drugs to market, potentially then provides the protective mechanism for the business, right? Or like, how do you think about competitive edge into the future if kind of you or others, you've talked about open sourcing, but there's plenty of companies that are out there being like, hey, we're going to deliver like a fully featured biological model that everybody can use. How does Insilico stay ahead of the curve within that context? Or how should any kind of AI drug discovery company try to like carve out competitive edge?
So when we are looking at competition and also where to learn from, again, we learn only from SpaceX, right? So we basically want to build SpaceX for drugs. And there, as I've explained already, the ultimate test is, does your drug work in the clinic? Does it help the patient? And there you're balancing novelty, confidence, commercial tractability, and many other properties, but mostly those three. And you need to demonstrate consistently that you can develop a preclinical candidate right from zero. So with various kind of levels of novelty of the targets and show consistency there, right? So you need to show that you can deliver payload to orbit. So we developed a set of benchmarks for that. We say we like last 5 years, 22 developmental candidates, clear cut. And many of those stories were published in top peer-reviewed journals like Nature Biotechnology. I have 2 examples from zero to developmental candidate. It shows you everything, how you do it, and shows you the experimental data. So the way to achieve leadership in this industry is to just be faster, cheaper, with higher probability of success. Actually, that's what most of the companies kind of have in their decks, but most did not deliver a single preclinical candidate. So it's kind of like folks that are competing with SpaceX, like blah, 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 look, we have a rocket. Oh, we have a better engine. We have a better fuel, a better simulation. We have better AI. Or we have the entire country supporting this effort, right? And then you look at how many did they launch over time and how many SpaceX launched. And you understand the industry, right? So if you are an investor, if you are a pharma company, if you are an employee, and if you are looking at how to rank the companies, the only way to rank them now is to say, okay, well, what's your average time to developmental candidate? Because that's where you're saving, right? The rest you are basically moving with the speed of traffic.
I understand exactly what you're saying, and it's kind of like. We don't know if the rockets have made it to orbit, right? It's almost like, in some ways you're arguing you should be judged on how many you launch. And there is validity to that in that kind of like the launch velocity is at the very least, you're learning something from it. And kind of like everything is going up or in launch mode, but we don't know how many have successfully made it to the orbit. Like you mentioned earlier, that kind of like pharma doesn't really like to basically buy novel targets because they rely on academia to do it. Because from their perspective, it's like, that's so far away from commercialization. How are they going to know this novel target's actually a target that's useful to do? They'll let somebody else take on that risk or an academic de-risk it before they even get involved. And similarly here, it's kind of, I can look at Insilico's pipeline and be like, well, yes, there's a lot that is, in IND enabling studies, but I don't know from an outside perspective, how do I know those are like actually, things that are going to
answer that. I have a great answer for you. So to do that, you need to sell a few of those assets. So once you get, let's say, to developmental candidate, if your level of quality is amazing, and if you did a really, really good job, usually, especially in the kind of moderate novelty target space or lower novelty target space, there is a market for them. So I've licensed out a few. So I basically licensed out in a very clear-cut way 3. So my most famous one was I took the drug to IND, and it sold. It got licensed to a company called Exelixis, one of the most hardcore medicinal chemistry companies that are discovering drugs themselves. They have billions of dollars in revenue from the drug they discovered and put to the market. So now they are discovering themselves and also buying from others. So they paid us $80 million cash up front for a molecule that just touched IND, touched phase 1, and it's $1 billion deal. Plus we have royalties. And then we've licensed 2 more at earlier stages, at preclinical candidate, right? One just got announced, $20 million up front. So it means that the level of quality of those launches are real, right? So basically,
what is that conversation like? Because usually it's hard to license out drugs at that earliest stage, right? I mean, you typically, like if you do the NPV analysis from a big pharma perspective, phase 1 is actually worth basically 0. It might be a net cost, given their rate of throughput from phase 1 to commercialization, the cost to bring something to commercialization. Again, depending on what market you're selling into, et cetera. But net, big pharma in some ways wants to just avoid all of that risk. So how does that conversation happen between you and a licensor where actually you manage to sell it in a cash accretive way? What are they seeing that really attracts them?
So usually that's why I said that the level of novelty must be moderate or moderate low. So we aim to be in a space when we are licensing out. We aim to be in a space where there is maybe 1 or 2 front runners. So it's not a crowded space, but people know the target. So they usually establish some trust in that target over time, right? So none of the targets are new, by the way. So the entire industry is dancing around like 30, 40 targets. That is funny. And everything else is kind of the variation on the old. So we don't like that game. We like to be in this, additional 300 target space or maybe more when we're out licensing. And there you're competing on quality. So we even had to go and learn from like Japanese biopharma, from German biopharma, how to do the data rooms, right? Because the quality of your asset has to be higher than the pharmaceutical companies got used to internally. Because internally they can say, oh, well, we didn't do this experiment, but we trust this hypothesis and we think that the molecule is going to work. That's why we're going to progress it forward. When they're buying, the level of due diligence is going to be much higher, right? Because if they usually have a few choices and they want to get the ultimately best one for the good price. And the price negotiations actually comes last, right? So they don't care about the price as much as they care about the quality of your data room. So when you are making molecules for sale, for licensing out, you need to make damn sure that they are better than the level of, I have a few papers here, right? So Nature Biotechnology can see the quality of our data rooms in some papers. So internally, when you're licensing out, it has to be higher than the level necessary to pass the peer review with 6 reviewers, right? Because there you're going to have 30 reviewers, and every one of them wants to kill the program, right? So it needs to be absolutely pristine. That's why I'm saying that it's basically like, imagine that you are doing a space launch, but you are also doing it to a US government agency that is going to bet like billions of dollars on some very expensive satellite, right? So they need to make sure that you are going to get there. And that's what's happening when you're out licensing. So you need to ensure that the level of quality is absolutely great. And if you are running with absolutely novel targets, right, that have never been in a clinic before, and I have one like that, and here is, a Nature Medicine paper that just came out. You can read up on how the clinical readouts look like and the quality of that data, and we disclose everything. So once you are going after a novel target, you would need to do much more work, right? Because pharma would want to partner with you after, let's say, phase 1 or preferably phase 2. So then they line up, they all want the asset, but then you are facing a different problem. You're like, okay, well, I have enough cash to progress it even further. And if I progress it even further, I'm going to get much more valuable data from the patients and from the study. I'm going to validate to the end. And if it works, it's billions of dollars I pay for the entire party many times over, right? And here is the punchline. So my job and my dream, so again, no claims, is to develop a profitable biotech. So if you look at how we operate, well, I can say that sometimes we're almost there because our revenue
What a
is
novel
coming
idea,
from,
a company that generates cash flow. I can't even believe it.
So because people got used to losing money, right? So my job, my kind of Mars mission for SpaceX is solving aging. Because if you solve aging, you solve everything else. You get enough time to solve everything else. And we change the society if we do that. But to solve aging, so far, there is zero successful companies in that field, right? So no drug works experimentally.
Okay, but see, this seems like a semantic issue to me because, and so now we're going to branch into the aging bit, or the longevity bit, because every disease is a disease of aging to some degree, right? And cardiovascular disease is a disease of aging.
Well, HIV is not a disease of aging, right? So HIV can be like any disease.
But it's aging plus environment. I just happen to be exposed to the wrong environmental variable, like that virus. I have all kinds of viruses swimming around in me. And, most of them, my body's like keeping under check because I'm like have a young, healthy immune system. But like, as my immune system gets like older and more degraded, then those viruses become like deadly viruses to me. That's kind of at least some of what's going on with cancer, I think. Or some cancers.
Well, not every disease is a disease of aging. Sorry, I have to correct you because like a huge variety of like pediatric diseases and some of the pretty painful ones are caused by, the mutations that you get before birth.
I think, we're just referring to kind of more complex common diseases. So, cancer on one side, like Alzheimer, Parkinson's, it could be just kind of the aging process actually happening in neurons. It's the kind of aging disease as well. And then like a cardiometabolic as well. It's like a tear of using your organism during the years, right?
Yeah, let me give you like a very tangible or specific example, right? There's a couple of gene editing companies going after a target that reduces basically bad cholesterol. I can't remember the name of the target, right? And there's
PCSK9,
Yeah. Well, there's another one, but yes, that's an example of it as well. You could imagine that, depending on how this works out, maybe modifying that, putting that mutation into the broader population actually reduces cardiovascular disease to a very broad extent. Is that separate and distinct from kind of like going after aging directly? Or do you think of those as like on a continuum of we're just basically like solving kind of these problems that happen to bodies as they age and or these different categories of thing?
So it's kind of the same and different. So you touched on a very, very sensitive topic, right? Because some things like, PCSK9, beautiful example, right? Most likely it's a longevity target. And the thing is, it's going to help a lot of people with bad cholesterol. But is it going to help people with good cholesterol? Good question, right? So on a population level, it may help, like GLP-1s, for example, right? GLP-1 drugs are very popular nowadays. You don't need to explain what it is. They are going to significantly improve, in my opinion, life expectancy in the US, right? Because here the percentage of the population that is obese is pretty high. But is it going to help you and me? Probably not, right? Because it causes muscle wasting. It increases the chances of age-related macular degeneration. We don't even know all the side effects, right? And by the way, your heart is a muscle. So probably for you and I, looking at your body composition, just I don't know how you look like outside. I'm making a joke. But most likely it's not going to help you, right? It's only going to, you're going to lose muscle. So we are looking for targets that would help you as well, right? So the idea is to identify the fundamental mechanisms that drive certain aging processes. Currently, what we have is called hallmarks of aging assessment, right? So there are certain buckets, certain types of biology that are at the same time associated with aging and driving aging. There is a framework in biogerontology that has been proposed only about 15 years ago that kind of classifies the different aging mechanisms. And what we usually do, we identify protein targets utilizing what we call aging clocks. So there's a model of human and animal aging where we see that the protein is specifically implicated in, it has some predictive value in predicting your chronological age or biological age. And then we also map those proteins that are implicated in aging clocks onto those hallmarks of aging. So we see in how many buckets of aging this protein plays. And then we also map it onto diseases. So we say, okay, well, now we have 3,000 age-related diseases. Where is it likely to work, this target, right? So you're basically looking for a Swiss Army knife. That it may work in as many as possible. And then you say, okay, well, which disease is most age-related, right? So where we can actually test this drug quickly, right? Where people die very quickly. So one of my favorite disease areas is idiopathic pulmonary fibrosis. It hits late in life. So usually average age of your typical patient would be 65, 68. And so they're reasonably on the older side. So they don't have a lot of life to live, and the quality of life is diminished. So you can run clinical trials a little bit faster and also collect, let's say, proteomic biomarkers to study the fibrotic signatures and antifibrotic effect of your drug, and at the same time use the same data to see if your drug is reversing the aging clocks.
Imagine there are like a number of, I would still argue they are artifacts of aging that are appearing in the body, but they're common across the body in some way. And then because they are happening common across the body, there's certain diseases that are triggered by enough degradation in that area. So idiopathic fibrosis of the lungs, pulmonary fibrosis is one that it basically like crosses a tipping point in people's bodies where something that turns on, it's really bad. So then if you can target kind of, you're really targeting what you think is that underlying process. And so then you're putting off this disease that otherwise would be triggered by the kind of like aging vector. And simultaneously, you're getting a measure of whether or not you can flatten or hopefully even like turn around the aging vector and turn it in reverse. Provocative. And don't you think that it's like the body is just composed of a whole series of independently evolved kind of interlocking systems that have probably evolved to like a similar kind of fail rate. And so that there's not, it's kind of like is it really likely that there's a shared common mechanism that is like kind of like driving the clock forward rather than, hey, it's like, this system breaks down at a certain like fail rate and this other system breaks down at a certain fail rate. And so you'd solve one, but you still have the others holding you back.
So I agree with you actually here 100%. So it is how aging transpires, right? So you basically have many, many different clocks ticking at different rates in different parts of your body at different level of organization, right? At the molecular level, at the cellular level, at the systems level, at the organ level, even at the psychological level, by the way. Right. So believe it or not, you are a different person now than you were, let's say, when you were 13, most likely. And by the way, I published a couple papers on that. I really did develop a deep learning model that tracks psychological aging, and that's something that you can reverse. I can basically for all your listeners, if you want to get younger while you're on this podcast, just religiously convince yourself that you are going to live to 120. Achievable, optimistic, and suddenly you are, if you're 60, you're halfway there. And it will change your life, right? You'll act as a younger individual. But don't feel sad if a lot of people who reverse their age this way, they kind of are not as happy because there is a long way to go. Sorry, jokes aside. So when you are trying to find this common ground, common mechanism that you can influence in order to slow down or reverse some aging processes, I think there are some. I believe that there are some mechanisms that are common. They are likely not to give you super dramatic benefit, right? So you might be able to get like 2 to 3 years. So some, like if you are using kind of your analogy, right? So imagine that we're talking about a car. So if you improve improve your brakes, right? Or improve something that is very easily breakable by reinforcing the material or fixing the damage, maybe the rest of your system, your engine can go for, properly maintained, it could go for hundreds of years. That's your brain, for example, right? And then you might need to fix the engine as well. But first you need to look at something that breaks the most, the fastest, and everywhere you test. So that's what we are trying to do, right? So we are trying to find those mechanisms that are more likely to give you additional 2 to 3 years, regardless of who you are, even if you don't have a disease.
But it's kind of like the car analogy is a good one because, I had a house that I put IKEA cabinets in and IKEA stuff in, and everything just fell apart at exactly like 7.5 years. Like that was it. And they all fell apart because they were all like engineered to like a certain lifespan. And don't you think like the body has evolved with, again, these kind of independent systems, but they're at some multiple above our procreation age, but not, 10x that they are all going to have some kind of breakdown mechanism at a similar kind of like clock
right.
distance.
I agree with you, right? So I actually don't think that the drug approach is going to let you escape aging. It's not going to happen. So, but I can tell you one thing, that if you get a drug that is safe and gives you everybody on the planet 1 year of quality life, you just generated 8.2 billion high quality life years. That is 170 million plus lifespans. So you basically just saved 170 million lifespans. And we're not talking about net present value. One drug, 1 year, right? So that's the impact I'm talking about. And I think so. So right now, just read this Nature Medicine paper that we published 20 days ago. It shows you kind of the path in the clinic, how you would do a clinical trial. And we published in that paper the proteomics data that people are now taking, so all over the world, and running aging clocks on that. So to see if we actually have reversed the aging signature. So I hope, and by the way, I don't recommend taking this drug, right? We are developing it for a hardcore disease. You need to take it at a high dose. It's not something that you would give to a healthy people or somebody with a moderate disease, right? It's designed for a hardcore disease. But we wanted to demonstrate a template that we use aging research as the source of a target. Then we do our own chemistry. We take it all the way using a traditional approach to an age-related disease. We collect the data that may be useful for aging research. And now we're opening this data up. So if you're an aging clock business, download, test, see if it works. We want to do it again and again and again and again.
Is the commercialization pathway, surely then there's, like you say, you have 170 million lifespans that you can, or what, I can't remember exactly what number you quoted, but And effectively, it's really the morbidity that kills you as much as the mortality, as in, like, you would rather, it's living to 121 versus 120 is not worth as much if that year is like a very miserable year, and actually living past 70, but then having degrading kind of quality of life is bad. You'd rather like put off all the aging related morbidity that occurs. And ultimately, isn't there a business in actually selling this to people as, hey, this is a life extension, or even not life extension, but a life improvement product? And can that work through the traditional commercial channels, or does it require a separate?
It's a great dream, right? So that is our Mars, right? So imagine if Insilico is SpaceX, our Mars is to be able to deliver two things. One is a super intelligent AI that understands aging in humans and all the mammalian species out there, so we can actually quickly test if necessary, right? To give mice our superpowers to see if they can actually catch up with us in terms of lifespans, or monkeys. And then we should be able to prompt this system to give us humans much longer lifespans, right? And I'm not talking about my current business model of selling drugs to pharma, right? So currently, this is to create a sustainable platform, right? So it's like Elon launching payload for others in order to generate sustainable business model to go to Mars, right? So our Mars is the superintelligence that will allow you to understand aging itself and very quickly perturb it. But at the same time, the molecules that we're putting out right now, right, in this papers, maybe this particular one, I wouldn't say that it's a perfect geroprotector, but we have several others that are following, right? That have very high potential to give you significantly improved quality of life in late life, right? So because you also, when you're 80, you lost a lot of function, right? So you're basically maybe operating at half of your kind of peak of your highest quality of life, right? Because you lost a lot of function. You cannot do the same things you did in a gym. You cannot do crazy things, right? In females, maybe they cannot reproduce as efficiently. You have different type of fun. So even if you restore some of this lost function, plus give a year, that's already a great achievement. Plus, we want to make sure that you first get the drugs approved for hardcore diseases, but then you expand the indications. So you develop the Swiss Army knife targets, Swiss Army knife drugs. You expand the indications to prophylactics, right? So for example, aspirin, great example. Aspirin, previously, we did not really consider it as a geroprotector, right? But now people over 65, take it as part of their tri-pill for, to reduce the probability of clots and other things, like baby aspirin. By the way, we still don't know if it works really well, right? So even though population statistics suggest that it does, imagine like over 100-year-old drug. So our job is to create more of those aspirin-like molecules that go after a disease, but then could be expanded into other things. And again, this gives you a platform to continue discovery. I'll tell you one fun story. So I founded a conference, and now I am a co-chair of a conference called ARDD, Aging Research and Drug Discovery. It happens every August in Copenhagen, 5 days. You must be there if you are in aging. By the way, I can for a high-profile investor.
Aren't we all in aging?
Well, we're passengers. If you want to be in a driver's seat, come to Copenhagen. So it's agingpharma.org. It is 5 days, 180 speakers, and we bring like maybe 40% are pharma and startups. I do it for 12 years. So out of those 12 years, I've seen companies come into this world with different business models, different drugs, different approaches. Somebody is doing a variation of already existing drug like rapamycin. Somebody is working on a completely new target, senolytics, xenomorphics. All those new modalities came out. all of them died, right? So, or dying, or no POC. You know why? Because even if some of them get significant amounts of money from, high net worth individuals or some visionary funds, most people don't understand how expensive biotech is, right? And how quickly you can burn if you are doing even one program. So unless you have a sustainable business model where you can very quickly validate your technology, make this technology available to the entire community, so you get community validation. That's why we sell software. And then you also demonstrate that you can sell your drugs to other companies, and those drugs are of ultra high quality, and you do it consistently, and you get revenue streams that are now recurring, right? Because you start getting milestones. So you put like a cow in somebody else's stable, the farmer grows the cow, gives you, pieces of meat while it's growing, or milk. And then once it starts milking,
yeah, maybe not
give
chopping
you
off pieces of a meat as the cow's alive. I think milk is good.
Sorry, let's say milk. We're talking milk. No cows. And by the way, no offense to anybody in India who likes cows too much. So we like when you are milking, they give you a percentage of the milk, right? But you don't need to grow the cow anymore, right? So you build the cow, you genetically engineered, quote unquote, the perfect cow, perfect bull, gave it to the farmer, cow for milk. Otherwise, you're going to get different kind of material. You start getting the milk, right? So that's the idea. We basically want to create a sustainable business model in order to do aging research at scale and go to Mars, right? To basically give you additional 15 years, 20 years,
okay.
right?
So yes. The SpaceX analogy is that they need Starlink to generate the cash flow to fund Mars. You can't actually go directly to Mars, but you can use the technology you are developing to get to Mars to actually generate a lot of terrestrial revenue and then send your robots to Mars to build the Mars colony.
Exactly.
And so we're all hopefully going to live to, maybe we just live to 123. Actually, maybe we just live to 100. We all live to 100 and it's just a healthier 100.
No, that's a bad goal to have. Yeah. 120, that's your minimum, please.
Oh, okay. Okay.
Don't have something stupid. Just
give me morbidity reduction more than, I'm not sure I want to know. Things are moving so quickly now. It's hard for me to imagine, to cast myself 80 years into the future. I think there's a lot of ways that, who knows, maybe we'll be uploading consciousnesses at that point. We don't need a biological body.
Most likely.
Okay.
For that, you will still need my molecules, right? Because you would still need to understand, how to play with the biology of the brain while you're uploading.
Okay. All right. Alex Z of Insilico Medicine, thanks so much for joining FYI, the For Your Innovation podcast. It's a pleasure having a conversation, and we wish you great and good luck.
Thank you. Luck is all we need. And your wonderful patron, Kathy Wood, is somebody who I deeply admire. And I think that she made the world better in so many ways. Thanks to her, you've got SpaceX, Tesla, and many other things. So I hope that you're going to stay on this pathway, and let's work together. Let's make aging great again.
Great. Okay. All right. Thank you. Well, what a fascinating conversation. Nemo, 5 years from now, will the maximum human life, will we have evidence that the maximum human lifespan is going to begin to extend by multiple years? Or do you think that this is actually not even 5, within 10 years, are we going to have line of sight to 128-year-olds or something?
In 10 years, I think yes. So this is the thesis, right? If the AI keeps kind of speeding up how it's doing now, I think that we can really see profound changes in the healthcare, and especially on the side of the kind of diagnostics. And so I think that in itself kind of be, kind of pushing more for actually understanding how much actually in the healthcare it's all about doing genomic sequencing, doing the testing, and how much is that actually kind of helps you with kind of preventive predictive medicine. And push for that is going to kind of push us more towards the aging and those kind of disorders. So that's how I see it.
See, I think like there is a future state in which it happens, but I think the problem is it really resolves back to the business model. As in him talking about all the backflips that he was doing to try to kind of like demonstrate that there's this, thing that he's potentially reversing within the aging chain, which I don't know, you all can read the papers and decide. And like he said, don't take the drug. It's like an experimental drug, et cetera, et cetera. And like if you had a world in which kind of people were peddling snake oil, like literally, like if instead of saying that, he was out there being like, for, $99 a month, you can try this thing out. And a lot of people were sold on doing it. And then as a result, a lot of people took a lot of things that actually didn't do that much for them and maybe were deleterious. But then we would have such massive experimentation that you would end up with some people, some large body of people getting, sold snake oil, which happened to actually work. And then you would learn that it worked and you would be like, oh my gosh, these people really are like, living forever, and so, and right now, the only basically example of that we have is the kind of like quirky billionaire types who are trying to do it to themselves. But it's not that many experiments. It's only a couple. Like we really need like experiment at scale. And really the only way you get to that is if you're like, go for it, guys, good luck. And accept that you're going to cause some like harm and some wasted resource along the way to get the data back that kind of gives you the true, like, hey, this is the way in which we're going to extend life. Because that set of market dynamics doesn't meaningfully exist, and nor is there like even a political entity that seems willing to let that exist. I think that just slows down our pace of progress towards this space where it's hard to kind of, if we're going to piecemeal our way there, where it's like, I'm going to solve this particular disease and as a byproduct show that this particular thing works, and then slowly try to expand and get it used off label, then it's out there before that problem is solved. So
yeah, this was today or yesterday. There was the news that Illumina acquired the SomaLogic from Standard BioTools, right? And blood-based kind of proteomics is like really going to be a huge market, but that's also the tool basically to actually understand the markers of aging. And so when you have actually the tool, so early, and first you need to have a tool to actually understand the kind of aging really, and then you can kind of go after it. And so just seeing how those things are developing kind of gives me the confidence.
Yeah, I mean, I guess that would be maybe you drive people into a state of anxiety where they're trying these experimental things because, oh my gosh, my biological age is actually like 55 instead of 45. And so I feel like I need to go after this because basically the clock is ticking. Maybe.
There was quite a few papers about aging, and I think the most prominent one is where, with the kind of immune system. So basically kind of your immune system is your true age. And so if your immune system is like not working great, you're not in a good condition. I would not be surprised that, that there's like therapies where basically kind of exchange your immune system or something like that. That people are trying some things like that.
Well, but again, I think it's like, yes, there's therapy. How do you successfully develop that kind of thing and then sell it in a way that people can buy it? I think is what it resolves to. As in, it's possible you can develop that, but the problem becomes like you can't do it in a way that demonstrates, yeah, this really extends life. You kind of have to have the buyer, and not even the patient, maybe that's the wrong term, the consumer, be like, yeah, I'm willing to take that risk. And right now, the whole system is set up where you get obscenely punished for trying to go about it in that way for good reason, because it's like, people buy supplements that clearly don't do anything for them and supplements that maybe they do. And because of that, you actually never get a true knowledge of whether or not they work. I don't know. It seems like a challenging space with, like he said, millions, hundreds of millions of life years that we could or lifespans that we could potentially save. And we'll see. Okay, well, thank you everybody for joining FYI, the For Your Innovation podcast. It's been a pleasure, Nemo, and I hope that your future comes true rather than mine. But here's an area where I am cynical and skeptical. Thank you for listening to this episode of FYI, the For Your Innovation podcast. If you enjoyed this episode, please hit that like button and make sure to leave a comment down below letting us know what you thought. And if you haven't already, please make sure to subscribe to the show and follow us at ARK Invest on your favorite social platforms so you stay up to date whenever we drop a new episode.
ARK believes that the information presented is accurate and was obtained from sources that ARK believes to be reliable. However, ARK does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARK. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARK's current views and assumptions, and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.